because I skipped over it last time, and I had to redeem myself. Elton is one of my favorite artists ever. So, okay. No worries, I'm redeemed. Oh, good. Okay. Whew. Thank goodness. All right. Cellular respiration. Let's take a look at an overview of cellular respiration. So last time and the time before, we've been talking about enzymes, we've been talking about energy, metabolic reactions, catabolic reactions, anabolic. Cellular respiration is truly the most important catabolic chemical process in our cells. And it is really this subsequent breaking down biochemical pathway. Remember, a pathway is like reaction after reaction after reaction. So it's this sequence of breaking apart glucose. So we start with glucose, and glucose is going to be continuously picked apart. Well, and we call it oxidation. It's continuously oxidized reaction after reaction, oxidation reaction after oxidation reaction. We're going to talk about what that means. To ultimately, at the end of all of this, the end of the story, when we finish next time, produce water and carbon dioxide. And of course, this process also requires oxygen. And what else we learned about a catabolic process, catabolic reaction, they are exergonic, right? When you pull apart these molecules and break them apart, they release energy. That's what exergonic means. And in the cells, when we're talking energy, we're referring to the energy currency molecule, the high energy molecule that we drew out, right? The adenosine triphosphate ATP. And so this is the broad overview. And mitochondrion, that's the organelle involved, at least for part of this. And we're gonna go into detail of how we do this, how we break apart glucose using oxygen to produce water, carbon dioxide, and energy. And of course, glucose is a sugar. We get it from food to get energy. We need energy, right? To do those processes we've mentioned, active transport, movement, etc. Okay, so let's go to the board. So first thing we're going to write is that cellular respiration occurs in 3 stages. Just got to get my chair out of the way here. Let's try to turn this lamp on. It's a little better. And let me stop sharing the screen, of course. All right, so cellular respiration is three stages. The first is called glycolysis. The second has a variety of names. It has 3 different names, but most commonly, the 2 most common names are the Krebs cycle, named after the guy who discovered it, Hans Krebs. This chair is in the way. Ugh, come on. There we go, that's better. Krebs cycle. And the third, ran out of room here. The third that we'll definitely be addressing next time is called the electron transport chain. So those are our 3 stages of cellular respiration. Also, Krebs cycle, another name that I might use interchangeably, same thing as the citric acid cycle. Okay, and then again, I'm just gonna write this so you have it in your notes. This is gonna be a catabolic process where we're breaking down glucose to produce water and carbon dioxide. So H2O and CO2. And we can even say this is catabolic because glucose is a larger molecule and water and CO2 are smaller. And then this, of course, also releases ATP molecules, making it an exergonic process. Move this up a bit. My neighbor plays terrible music sometimes. Yeah, I don't personally like it. I had to shut my window. Okay. Now, the next, before we dive in, we have to address this concept of oxidation, what it means to oxidize a molecule, and reduction, what it means to reduce a molecule. So oxidation and reduction, or sometimes we call it redox reactions, redox chemistry. So that's what we got to talk about. So let me go to the next slide. So this is oxidation and reduction. Okay. So as you can see, oxidation refers what it means to oxidize molecule A here. What it means to oxidize molecule A is to strip an electron or two electrons, multiple electrons, whatever. It's one or two really. To strip electrons from the molecule. Remove them. You oxidize the molecule. That's what it means. You're taking electrons from it. So if you oxidize A, you lost a negatively charged electron, right? So it has this positive charge now. And then B, if we were to give it an electron, this molecule, you actually reduce the molecule. You gave it a negatively charged, remember electrons are negatively charged. You gave it a negatively charged particle, and so you've reduced it. And it has that negative now. You gave it an electron. Okay. So in biology, when you learn about this in chemistry, they really like their electrons. But in biology, we're going to, of course, mention electrons, but we also track hydrogens, hydrogen atoms that actually carry these electrons with them. And so when we look at oxidation, and when we look at reduction, oxidation was the loss of an electron, right? We can also say oxidation is the loss of hydrogen, of a hydrogen atom or two hydrogen atoms, whatever. And the electrons will be with that hydrogen, actually the hydrogen will carry the electrons. But I really want you to pay attention to hydrogen. So if I oxidize a molecule, I remove hydrogens from it. If I reduce a molecule, I will give it. It will gain hydrogen. So this would be the gain of hydrogen. So I'm actually going to rewrite the chemical formula for cellular respiration, which is glucose. This is the chemical formula for the glucose molecule. Glucose plus oxygen molecules, 6 oxygen molecules, going to 6 carbon dioxide molecules and 6 water molecules. Man, it's way too hot in here. Okay. That's better. Get some air. It's like the music's off, so. All right, now, if I were to look at this carbon-based glucose molecule, before and then after the whole process here, would you call it oxidized or reduced? Did we oxidize glucose or reduce it? If we're going from glucose to carbon dioxide. Yeah, we oxidized it, exactly. Because there's no hydrogens, right? So this is oxidation, absolutely. You've stripped off hydrogens. Now, what about the, let's use green. What about the oxygen? I told you the oxygen is going to become water. Did we reduce oxygen or oxidize it? Did it gain hydrogens or lose them? Yeah, we reduced the oxygen to form water, absolutely. So this is reduction. Okay. And a fun way to remember this, there's a few actually. There's oil rig, and so oil rig oxidation is loss of hydrogens and electrons. Reduction is gain of electrons or hydrogens. Another funny one is Leo the lion goes grr. Leo, loss of electrons, oxidation. The lion goes grr. Gain is reduction. So, whichever you want to use to remember. But the way I kind of remember it is if you're reducing something, you're giving it negatively charged electrons, so you're reducing, right? Lowering its charge. And the hydrogens are always with the electrons, they're bound together. Okay. The next thing we're going to talk about is something I mentioned last time very briefly, which are coenzymes. So I want you to read that bullet point while I erase here. So last time we talked about coenzymes and cofactors being involved in chemical reactions by assisting enzyme function in some way. Cofactors were helping substrates bind, and really we're going to learn here why coenzymes, namely these two NAD plus and FAD, assist reactions of cellular respiration by acting as like these taxi cabs. Now, okay, before we really kind of see the details, I want to show you what I mean by it being a taxi cab type molecule, but they hold electrons, and the electrons would be our passengers in this analogy. So I really do like this analogy where the coenzymes are these taxis and they can pick up electrons and hydrogen, thus being reduced, right? And be full taxi molecules, or they can drop off their passengers, which are the electrons in our analogy, or hydrogens, both really, and be oxidized. So let's take a look at what that looks like. The full taxi, okay, when NAD is a prime example to use. When NAD plus, this coenzyme has electrons bound to it. It has passengers, it's a full taxi. It looks like this. It's actually NADH. It's NADH, and I drew the covalent bond to the hydrogen here on purpose. So it's our covalent bond, which is our pair of shared electrons, right? Pair of shared electrons. Now I'm going to demonstrate what it means to oxidize a molecule, namely this NADH molecule. So let's do an oxidation reaction. So this will be, what did I use? Blue. Oxidation. Let's oxidize NADH. If I'm going to oxidize NADH, I'm going to strip off the hydrogen and electrons as well. And so when I do that, it looks like this. It becomes NAD with a plus charge plus 2 electrons, so 2E minus, plus the H, right? Which has a plus charge. So this would be a proton or hydrogen ion, positive hydrogen ion, either way. So when I oxidize a molecule, any molecule during cellular respiration, I remove hydrogen and I remove electrons with it, just like that. And sometimes if you're watching like Khan Academy, they kind of show this as hydride, where the electrons are bound to the hydrogen. It has a negative charge. But I separate it out because I think most textbooks do that. But I do believe in reality that they are attached, but either way. It does emphasize that we're removing electrons, right? And hydrogen. Now, the electrons and the hydrogen are our passengers, right? So the NAD plus is the empty taxi. So NAD plus is our empty taxi. And I believe I have a cool picture of that. There you go. NAD plus. Now, if well, before I get to that, full taxi, which we started with, I'm just going to do the reverse reaction here. The reverse, where I reduce NAD plus by adding electrons and hydrogen, giving it passengers. So let's do the reduction reaction. Then you form NADH. And NADH is our full taxi. And then here's the picture of that. So here's our full taxi now. And we call it NADH when it has the hydrogen and the 2 electrons bound to the NAD molecule. NAD and FAD, they're just these giant carbon-based organic molecules derived from vitamin B, I believe. They act as coenzymes, and we'll see why that is. Okay. Oh, question. Yeah, so because the 2 electrons are being added to the empty NAD plus, and then now it's NADH, you basically jammed them together, and now they're bonded together with those electrons. Then you filled up the empty taxi, yeah. So let's practice this concept. So number 1, so determine if it's oxidation or reduction. NAD plus 2 electrons and hydrogen ion to NADH. Is that oxidizing or reducing? Yeah, it is reduction. Yeah. So we did that we did that here. Right. We did the reverse here. So it's reducing, right? NAD plus is gaining electrons and hydrogen, right? To be NADH. So when it gains it, if you add on a hydrogen or electrons, you have a reduction, right? Now, what about number 2? What's happening in number 2? Yes, oxidation. And that's because this time, instead of adding hydrogens and electrons to the molecule, we're removing them, right? We strip them off. FADH2 becomes FAD without hydrogens and electrons. We pulled them off, and that's oxidation. Cool. Now that we did that, we can get into this thing. By starting with glycolysis. Glycolysis, that's the first stage of this story. Okay, so let's write well, there's our glucose molecule, okay? So that's what we're gonna start with. But before we even begin the process, let's break down the word glycolysis. Glycolysis. So glyco refers to sugar, kind of like glycogen. We learned was that polysaccharide that we store in muscle and liver. And then lysis, we talked about the lysosome and hydrolysis, right? Who remembers what lysis means? What was lysis? To lyse something. Yeah, to break it or split it apart, absolutely. So what we're doing in glycolysis is we're breaking apart sugar, namely glucose. So we start with glucose, which I'm just going to draw as simplified here as this 6-carbon molecule. So this is our glucose. And so at the introductory level, we don't do all the steps of glycolysis. I mean, really, you only do that in graduate level biochemistry because there's 10 enzyme catalyzed steps, 10 reactions. So I'm going to simplify things here by going from the starting reactant to our ending reactant, two products. So there are 2 3-carbon molecules called pyruvate. Each one of these is a pyruvate molecule. So we end up with 2 pyruvates. 2 pyruvate. And a pyruvate molecule, it's a 3-carbon molecule, and we end up with 2. So we split the glucose in half. By the end of this, like I said, it's 10 steps, 10 enzyme catalyzed steps. Now, there are 2 other important occurrences during glycolysis that we do need to talk about that occur actually during these steps. Now, throughout this entire series of reactions, it's kind of like an assembly line where glucose is moving down. Oh, what does the word next to 2 say? Pyruvate. Pyruvate. It's P-Y-R-U-V-A-T-E. Yeah. So glucose is kind of moving down this assembly line. And what I mean is, this is a biochemical pathway. So when I say there's 10 enzyme catalyzed steps, I mean glucose does a chemical reaction, gets turned to a product. It's actually glucose 6-phosphate. Then that goes through another reaction, becomes another product. I believe it's 1,6-bisphosphate. Oh, I'm forgetting fructose 6-phosphate. Anyway, continuously modified, right? Kind of like it's going down this assembly line and being worked on. Reactant to product with an enzyme involved, right? Chemical reaction after chemical reaction. And so after the 10 chemical reactions, you get these two pyruvate molecules, the splitting. Now, during 2 of these steps, we're going to generate 2 NADH molecules. And I'm going to show you what that looks like. So for 2 of these steps, we actually oxidize the substrate, whatever it is. There's 2 oxidation steps. I think, what is it? I don't remember the specific steps of glycolysis, but essentially glucose becomes another molecule, right, as it's being modified throughout these 10 steps. And then 2 of these steps, that molecule is going to be oxidized with the help of NAD. So NAD plus is going to come in as a coenzyme with the enzyme involved in these 2 steps, 2 different enzymes. But NAD plus is going to help oxidize the substrate in this pathway. So it's going to strip off electrons and hydrogen, as we just kind of went over, to form NADH. And again, this happens twice, so 2 times. So we end up with 2 NADH molecules here. So I'm looking it up because I want to know. Glyceraldehyde 3-phosphate. Okay. That's the substrate that gets oxidized. NAD plus becomes NADH. Okay, sick. So that happens twice. And the other step, don't worry about it. I'm just kind of, again, this is intro, so we're not going to go too crazy here. 2 other steps, we produce ATP, actually. So we do get a little bit of energy from glycolysis. And so what happens is for 2 of these steps, we're actually going to also take ADP and stick on a phosphate to form ATP. And again, this happens twice. So 2, 2. Okay, so by the end of glycolysis, yes, it is inorganic phosphate. Yep. So by the end of glycolysis, we end up with 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules. 2 other details here, just description, where it occurs. Where does glycolysis happen? It happens in the cytoplasm. So it occurs in the cytoplasm of the cell. And this is actually for both prokaryotes and eukaryotes. And since I said both prokaryotes and eukaryotes, we learned back when we talked about cells, that's everything on the planet. Glycolysis is the most ancient, primitive energy pathway. And the first cells billions of years ago were doing glycolysis to produce ATP. But it occurs in cytoplasm. And if you'll notice, oxygen has not played a role yet, so it does not require oxygen. It's an anaerobic type pathway. So it does not require oxygen. Those are the only other details that I want you to know for glycolysis. Okay. Sorry, my phone is acting up. Here we go. Okay. Now, we have to answer a question. Oh, by the end of those 2 steps, the 2-pyruvate, correct. But it's not 2 steps, it's 10, right? So after all 10 steps of modifying glucose, we end up with the 2 pyruvates, 2 NADH molecules, and 2 ATPs. Okay, now, I'm sure some of you have heard the word fermentation. And to ferment or not to ferment is a major decision junction point for a lot of cells. So we're going to talk about what that means. Okay, like I said, all cells do glycolysis, us included, right? And so all cells are going to start with glucose. And it's going to undergo glycolysis, which is our first stage, right? Glycolysis. And I'm just going to add in here just to remind us, and it'll be relevant soon, that glycolysis gives us 2 ATP molecules. And we convert the glucose to 2 pyruvate molecules. Okay. Now, after cells do that, all cells, there's a decision point. And it kind of depends on the organism and the cell type and the conditions, namely. And we're going to start with fermentation. And when a cell that's able to ferment will perform fermentation. And that's when there's no oxygen around. So in an anaerobic environment where oxygen is lacking, the cell performs fermentation, which I'll do down here. I'm also going to write oxygen is absent. And the fancy word for that would be anaerobic. but I'm going to write oxygen is absent. So this is an anaerobic process. Okay, so depending on the organism, there's two different types of fermentation. It's actually kind of interesting. So we'll start with my favorite, alcohol fermentation. I like beer. So alcohol fermentation, so this is producing wine plus beer. This is performed by yeast cells, which is a type of fungus. These are single-celled fungal organisms. So let me do, I don't know why I wrote wine and beer, but whatever. Well, yeast is used, right, to produce wine and beer. So I'll write examples are yeast. There's certain bacteria that do it too, but it's mainly yeast. It's also, interestingly enough, alcohol fermentation, you don't have to know this, it's just kind of interesting, actually produces CO2 as a byproduct, gas. And so baker's yeast that helps dough rise, they'll ferment and produce alcohol, which evaporates. That's why you don't get drunk if you eat pizza or have bread. But the CO2 causes the bread to rise when they ferment and produce alcohol. The other type that we do, as well as most bacteria, is called lactic acid fermentation. And since I wrote wine and beer, this is used to produce a lot of yogurt, like Greek yogurt, certain cheeses. And the organisms doing this are certain strains of bacteria. And even, might ask about this on the exam, I feel like I had a question I've used before about this. But our muscle cells ferment and produce lactic acid during strenuous exercise. The reason being, during strenuous exercise, if you need ATP, actually, before I say that, let me just back up and say that this anaerobic pathway where organisms will ferment pyruvate and produce alcohol or lactic acid happens without oxygen, right? Anaerobic. This whole thing produces 2 ATP molecules from glycolysis, right? That's it. Kind of not that great, right? So fermentation is really just kind of, I like to call it the quick, dirty, cheap, easy way to make energy. It's quick, it's easy, but it's not quality, it's not really producing that much. It's just 2 ATPs from glycolysis by fermenting pyruvate. Now, if you're wondering why ferment pyruvate at all, you already got the ATP. You don't have to know that. But essentially, fermentation allows glycolysis to happen again. So it kind of reloads the glycolysis gun to produce another 2 ATP. So fermenting allows this process to repeat. But it's not very efficient, is it? Producing 2 ATP. And so our muscle cells, back to the muscle cells, they only resort to this when it has to. Strenuous exercise is when we ferment and produce lactic acid. when oxygen is not enough. We need more energy, and the oxygen, that pathway it's not fast enough, the oxygen pathway. We need it now because we're running really fast. We still need energy, so it's going to ferment the muscle cells. So they've kind of adapted to do that. It's pretty fascinating. Okay, did I see a question? So Anastasia, that's a great question. I actually was just reading about this. They used to think lactic acid was the reason for muscle soreness, but I think recent research, not I think, I just read it, recent research actually demonstrated that to be false. The reason for soreness, so the lactic acid, it was not causing muscle soreness. In fact, I believe they said lactic acid actually stimulated growth and it was good, but the reason for soreness is inflammation, namely, and tearing the muscle cells apart is going to cause an inflammatory reaction with pain, and so there's going to be damage associated with that. That's what the recent evidence was suggesting. Yeah, Sophia, so that's correct. Yeah. We need it energy, we need it and we need it fast. Car alarm, great. Anyways, but as a segue, send the article to us. I will actually, but the funny thing is it's not an article. I just read it in our textbook. I like to read the textbook because I get, I'm lucky because I'm the instructor. I get a free instructor textbook and it's the latest edition. So it has recent research in it. But yeah, I can take a picture if I can find it again. I think I'm on another chapter, but yeah. Why don't I write that down? Yeah, I'll find it. Where was it? Oh, well, it's definitely in the respiration chapter. So yeah, I'll take a picture. Yeah, it's pretty interesting. And I'll send it to you guys. I like that you're interested. That's cool. All right. Now, as a segue, let's do the opposite. Let's do something that's much more efficient, that's going to give us much more energy, that our body prefers, that most multicellular life prefers. The oxygen pathway, which is the focus of the rest of today's lecture and next time. So when oxygen is present, the aerobic pathway. This is aerobic, right? So this, we don't do fermentation. Organisms don't do fermentation that do this. These organisms, including us, do aerobic cellular respiration, which, kind of ran out of room there. Aerobic cellular respiration, which is, as we discussed, glycolysis, and then the Krebs cycle, which if we have time today, we'll talk about. What time is it? Oh, wow, we have plenty of time. Dang, cool. Krebs cycle, and I'll just abbreviate it here, because I wrote it already, and I abbreviate it anyway a lot. The electron transport chain, ETC. And this process, including the 2-ATP from glycolysis, is going to produce, depending on the organism, 36 to 38 ATP molecules. So it's a lot better, a lot more. This question. Aerobic cellular respiration. Aerobic cellular respiration. Actually, as it gets darker here, I can move my lamp. Did that yesterday for my other class. I should get like a light that just kind of like shines on the board. That'd be cool. Maybe just move my lamps. Well, that's working. All right. Now, the next thing I'm going to do, so I mentioned that glycolysis occurs in the cytoplasm, right? The fluid of the cell just floating around. Right, so glucose is broken apart into 2 pyruvates in the cytoplasm. But now that we're going to discuss this aerobic pathway, and we're going to talk about Krebs cycle, the pyruvate's actually going to move into a very important organelle for energy production, namely aerobic energy production, the mitochondria. And to get a sense of where things are happening, we're gonna draw one. We're gonna draw a single mitochondrion. So let's do that. And here's I believe a electron micrograph of a mitochondrion. We learned about the mitochondria when we talked about organelles. Okay, and let's see. So we're gonna kind of recreate the mitochondrion because its morphology, meaning its cellular shape and architecture, is important with where things are happening. So it's this bean-shaped eukaryotic organelle, right? Kind of like that, kind of. And interestingly enough, it has 2 membranes. So this is its outer membrane. Right? And then this is its inner membrane. And the inner membrane is all squiggly and consists of these folds. So this is the inner membrane. And just because there's not enough fancy biology words, the folds, we actually call them cristae. So the folds of the inner membrane are called cristae. Which is C-R-I-S-T-A-E. I think a single one is a crista, but okay, so double membrane. And then the center of the mitochondria here is called the matrix. So the area in the center is called the matrix. And then the space in between the membranes, this space is called appropriately, the intermembrane space. So I'll write that. Let me make some more room for myself here. This is the intermembrane space, which is in between the two, the outer and the inner membrane. Okay, so importantly, the remainder of our aerobic cellular respiration occurs in the mitochondrion. But glycolysis occurred out here in the cytoplasm, right? So this is where glycolysis happened. And then the two pyruvates are going to be shuttled into via transporters, protein transporters in these membranes. Pyruvate's going to go directly into the matrix with Keanu Reeves. And it's a lame joke, but that's where the Krebs cycle occurs, in the matrix. Krebs cycle. And then our electron transport chain, which we're going to tackle next time, actually occurs along the inner membrane. And so I'm going to draw some boxes, little rectangles to represent proteins of our electron transport chain. It actually occurs across the inner membrane, but we'll explore that in detail next time for sure. Oh, got some LOLs, nice. Okay, so with the last 10 minutes, no, 15 minutes, we're gonna tackle it. We're gonna do the Krebs cycle in a simplified version. ETC, that's all I wrote. It's the ETC. It just occurs along this inner membrane, or across it actually. But we'll see that better next time. Okay. So the Krebs cycle. Hold on. I had to shut my window again. Then it gets too hot. Ugh. All right. Well, it's 15 minutes left, so I'll endure. All right, Krebs cycle. We're gonna do it. It's kind of crazy. Let's go for it. And actually, there's a small caveat to the Krebs cycle. Something happens right before it. Sometimes separated into another stage, but it's really part of before we can enter the Krebs cycle, and it's going to happen in the matrix. And it's called pyruvate oxidation. It actually has several names depending on your textbook. Pyruvate oxidation is one name. It's also sometimes called the intermediate step, or the transition step, or pyruvate processing. I typically use pyruvate oxidation. So let's do that. So let me first. Okay, so steps 2, A and B, I kind of separated them out. So the pyruvate oxidation and the Krebs cycle. So step 2A will be pyruvate oxidation. So let's do that. So 2A. Now, as we continue our process here of modifying and reacting glucose subsequently and converting it to different substrates and products, we finished off glycolysis with 2 pyruvate molecules. So I'm going to draw a simple pyruvate molecule to start this process. So it was a 3-carbon molecule, and it actually has this double bond oxygen with another oxygen single bonded at the end. That's all I want you to know. I'll show you why that's important. So this is pyruvate. And yes, there's 2 of them, but we'll get to that. First, we'll just do one at a time. Okay. So in a similar manner, there's going to be a few reactions. I believe there's 2 during processing or oxidation of pyruvate. But we're just going to go to that end product, which actually has 2 carbons. And it binds, or it's bonded to a coenzyme called coenzyme A. The squiggly means it's coenzyme's like a bigger molecule, but you don't have to know that. But it's coenzyme A. This 2-carbon molecule attached to coenzyme A is called acetyl-CoA. So this is acetyl-CoA. That's right. So pyruvate becomes acetyl-CoA. The function of coenzyme A is merely to bring the 2 carbons to the Krebs cycle. Don't worry about that. Okay. During pyruvate oxidation, like it implies, we're going to oxidize pyruvate. So what that means is we're going to strip off electrons and hydrogen, right? As we learned, and form another NADH. So NAD plus 2 electrons plus the hydrogen ion becomes NADH. And what also happens during pyruvate oxidation, so during another enzyme catalyzed step, this carbon gets cleaved off. As we noticed, we end up with 2 carbons, right? So we lost 1. So I'll do blue. So I'm going to break that bond, right? What is this molecule? What am I going to release here? What's going to come off? Yeah, CO2, it's carbon dioxide. So you form a carbon dioxide molecule, which is released from pyruvate and during this process. Okay. Now, that's all it is. But I mentioned, of course, and we know we ended up with 2 pyruvates after glycolysis. So, of course, this means this process happens 2 times. And if it happens 2 times, we're going to make 2 NADHs, right? Total. 2 acetyl-CoAs, and then 2 CO2s. looks funky, but let me erase that. 2 CO2 molecules. Okay, so that's the pyruvate processing 2A step. And now that we have acetyl-CoA, acetyl-CoA is able to enter the Krebs cycle to begin 2B. And so I usually have a giant whiteboard, but I think I'm just going to erase this. But we're going to start with this 2-carbon molecule called acetyl-CoA in the Krebs cycle. So let's do that. So this would be 2B. So let's do the Krebs cycle. And just keep in the back of your mind that we have 2 acetyl-CoAs total, right? But let's just do one cycle for now, and then we'll take a look at what really happens, which is the cycle occurs twice. But let's take one of those acetyl-CoAs and do this Krebs cycle and plug it into the Krebs cycle. So it's this 2-carbon acetyl-CoA, so it has that CoA. The CoA leaves, and the first thing that happens in the Krebs cycle is that the 2 carbons attach to this first substrate of sorts, or first reactant in the Krebs cycle, which has 4 carbons, called oxaloacetate. So this acetyl group is going to combine with oxaloacetate. That's the first step of the Krebs cycle. And so now that has 6 carbons, right? 1, 2, 3, 4. Okay, so there you go. 1, 2, 3, 4, 5, 6. Draw that arrow better. So that combination is the first step. And this 6-carbon molecule is actually called citrate. Hence why sometimes the cycle is called the citric acid cycle. Citrate, citric acid, they're actually pretty much the same molecule. Don't have to worry about that though. Okay. Now, the Krebs cycle is kind of like glycolysis in the sense of there's a lot of steps, but we're going to simplify it. I believe there's 8 steps, 7 or 8. And just like glycolysis, we're going to be producing these products. There's going to be molecules formed. And so to keep it simple, it's called a cycle because oxaloacetate gets regenerated at the end so that another acetyl-CoA can combine and can do the cycle again. But like I said, that's the first step. After that, citrate gets converted to alpha-ketoglutarate, gets converted to malate, fumarate, succinyl-CoA. There's a lot of steps. With all these steps, we're going to be producing molecules, just like glycolysis. And what we produce for one cycle is an ATP. Do that in another color, keep things interesting. 3 NADH molecules and 1 FADH2 molecule, our other electron carrier taxi molecule. And then there's also CO2 released from the Krebs cycle. Per cycle, 2 CO2 molecules. In fact, in regenerating oxaloacetate, you lose 2 carbons, right? So I'm gonna cleave off 2 of these carbons, which is going to make 2 CO2 molecules. Okay, thing for our last minute here, we got to multiply everything by 2. Reason being is we're going to do the cycle twice. For each molecule of glucose, we do the Krebs cycle 2 times because from pyruvate oxidation, we ended up with 2 acetyl-CoAs, right? So we do this, the citric acid cycle, Krebs cycle 2 times. So total, we get 2 ATP, 6 NADH, 2 FADH2, and 4 CO2 total, right? Because each time we got 2 of them, so that's 4 total, 4 CO2 total. By the end of all this, we have completely oxidized glucose. Glucose has become carbon dioxide molecules. The glucose that we started with. We have stripped off hydrogens and electrons from it, and it has now become completely broken apart into CO2 molecules, completely oxidized. Okay, that's it. Did I see chat? Why does acetyl-CoA have a squiggle? Basically because the CoA is like a giant coenzyme molecule, that's all. It's just implying that there's more to the CoA, but you don't have to worry about that. After glycolysis, would it have 6 NADH? So after glycolysis, there's 2 NADH. Oh, I get what you're saying. I get what you're saying. Great question. Glycolysis, 2 NADH, right? And we'll kind of review this at the beginning of next class. 2 NADH from glycolysis. We got 2 NADH from the pyruvate oxidation. So now we're at 4. And then we got 6 from the Krebs cycle. 2 times, right? 10 NADHs total by the end so far, right? That's actually as much as we're going to get because we've completely oxidized glucose now. Okay, I feel like I'm over time, so let's call it there. Other questions? Lab exam questions, lecture questions, anything? This is pretty complex stuff, I know, so. Nothing, huh? Oh, okay. Yeah, you can send me your journal with formatting. Sure. I can give you some tips on the formatting, some quick, like, hey, be sure to do the APA style format and stuff with citations and whatnot. Kelly, so I would say both. Both, for sure. Some are directly with the results, like what certain colors would indicate for biological molecule identifying, right? Or if the solution's acidic or basic from the pH lab. And then some conceptual stuff too. Trying to remember off the top of my head. I made it yesterday. There's definitely conceptual as well. Let me think. We had. So like enzyme, that reaction, and you know what's happening in that reaction. So that's a bit conceptual, right? Yeah, fluorescence, sure. Yeah, you can do that for a subtopic and go into sort of what is fluorescence? What does it mean for a molecule to fluoresce? Sure. Yeah, get into the physics of it a bit. Sounds cool to me. Sure. Yeah, illustrations don't have to be from your primary source. No. You can use one or something, but yeah, no. You need at least 3, right? So it doesn't have to be from the primary source. The primary source I suggested, pubmed.gov. If you type in bioluminescence, Glorianne, for example, there should be a good amount of research papers that are primary source literature for you to use. Unfortunately, if you're on MiraCosta's Wi-Fi, the school pays for some of those journal subscriptions, so you get more access. But there's a good amount of free info as well. Yes, that's the one, pubmed.gov. Coolio. Well, good luck on your lab exam tomorrow. Just email me if any of you have more questions, unless there's anything else right now. And have a great weekend too. Alrighty. Thanks, guys.